Okay, hello everybody. So this is a podcasted lecture of the DNA damage and repair session from Molecular Genetics, Year 2 Biomed. So I want to start the lecture really with a couple of quotes to try and put in context how important DNA damage or the repair of DNA damage is to a cell, to an organism. So we'll start with a quote by Francis Crick, who said that DNA is in fact so precious and so fragile that we now know that the cell has evolved a whole variety of repair mechanisms to protect its DNA from assaults by radiation, chemicals and other hazards. This is exactly the sort of thing that the process of evolution by natural selection would lead us to expect. And then a second quote by a more modern, if you like, or recent scientist, Professor Steve Jackson. This guy's a leading expert, a world expert on DNA damage repair. And this is a really interesting, I think, well, astounding fact. And what Steve Jackson says is that the amazing thing is that we have around 10 to the 14 cells in our bodies. And every cell in every body is sustaining around 100,000 DNA damage events or lesions per day. So if you think about that, our bodies, even if you're not sitting in the sun or exposing yourself to dangerous chemicals in a lab, etc., your body's still undergoing a huge amount of DNA damage, somewhere in the region of 10 to the 18 to 10 to the 19 independent DNA damage events per day. So that's a huge number. We're looking at 10 million DNA damage events which are occurring in everybody sat in this lecture theatre every day, body-wide here. Now if one of those DNA damage events goes unrepaired, undetected, in theory that can lead to transformation and ultimately to cancer. So how on earth are we able to cope with such a humongous amount of DNA damage assaulting our bodies? How is that actually kept up with? How do we monitor that? How do we repair it? It seems such an unfeasibly large amount to deal with accurately and with fidelity. So that's what we're going to talk about today. The lecture outline is we're going to discuss the different causes of DNA damage initially, so things which lead to damage of our DNA. And we'll talk then how such damage is detected and repaired. So these two processes are obviously as a whole are important, but it's important to realise that DNA damage repair mechanisms need to be able to detect the damage in the first place and then go about actually repairing the damage. And we're going to consider different mechanisms from both prokaryotic and eukaryotic systems. E. coli bacteria again provided an excellent model organism from which to study DNA damage and DNA repair, and a lot of the systems actually which exist in bacteria and E. coli, for example, also exist in us. And the idea is that by the end of the lecture and by the time you've engaged with some wider reading, you should be able to describe several types of DNA damage and detail how such damage is detected and repaired by the cell. So an excellent source of reading material for this lecture is the book Molecular Biology of the Gene by Watson et al. This is on the recommended reading list for the module. I would also strongly recommend this book for the level 3 module Medical Genetics. So anything molecular biology related, I would say this book's as good as anything, to be honest. So if you don't own the book, it might be worth thinking about purchasing it. It's got an excellent chapter on this lecture and some of the other lectures that appear on this module. Okay then, so how exactly does DNA damage occur? Well, there are two broad categories that we can put DNA damage events into. The first is intrinsic DNA damage and the second extrinsic. So the vast majority of DNA damage actually occurs during DNA replication. So there's an innate amount of error that occurs during replication of DNA, and we'll talk about that in a moment. But there are also kind of these naturally occurring compounds and things which are also able to damage your DNA. This is environmental in nature, but it's largely unavoidable. We can't avoid this type of DNA damage, and together replication errors, hydrolytic and reactive oxygen species damage represents this intrinsic category of DNA damage agents. We also have then the extrinsic DNA damaging agents. These are perhaps the agents which you've been most familiar with or that would come to mind initially when you thought of DNA damaging compounds, and this includes radiation and these mutagenic chemicals, which we'll talk about in more detail later on. So if that's how you damage DNA, if they're the processes which damage the DNA, what's the actual consequence of damaging DNA? Well, there are two obvious consequences. The first is the introduction of mutation to the DNA sequence. And the second, which you may not have thought of as important, is a structural alteration to the actual DNA itself. So by damaging the DNA, you're either going to lead to the establishment of mutations in the DNA sequence, which is obviously problematic, or in the short term you may introduce structural alterations to the DNA, which is problematic for the cell as well, and we'll discuss why that's the case throughout the lecture. So in the case of an introduction of mutations then, if this is a parent molecule of DNA that's undergoing replication, this double-stranded DNA molecule is replicated to give two further double-stranded DNA molecules. In the first instance here, everything's fine. In the other daughter cell, there's been a replication error. So rather than introducing a thymine opposite this adenine here, we've introduced a cytosine, a C. So there's been an error in the actual fidelity of the DNA replication process. Now, as things stand, that's not a massive issue. This is a transient change. It's not become established yet as a mutation in the actual DNA sequence. What we've got is a mispairing event. However, if this isn't detected and repaired before the next round of replication, what happens then is one of these strands acts as a template for the granddaughter double-stranded DNA molecule, and the other strand, which contains this incorrectly inserted base, acts as a template for the other granddaughter DNA molecule. And in this instance then, when DNA replication occurs, this C, which shouldn't be there, is used as a template, and then you get an introduction of an actual mutation into your DNA. So one of the key things about repairing replication errors to mutation that occur during the DNA replication process is that they need to be detected relatively rapidly, because if they aren't, then they can become established as mutations in the actual gene or the DNA of the individual. The second consequence here, this structural alteration, well, that's particularly important during replication, but also during transcription of DNA. So transcription is a process by which DNA is converted into mRNA, which is then in turn translated into protein. So during transcription, particularly in higher organisms, there's a large group of proteins which come together around RNA polymerase II, and then RNA polymerase II starts about the business of copying the DNA into the RNA. However, if there's a structural alteration to the DNA that RNA polymerase II is attempting to transcribe, that can cause this RNA polymerase II complex to stall during transcription. So it reaches this bulky adduct here, this change in the structure of the DNA, and it's unable to get past that, and that causes transcription to cease, and obviously can lead to major problems, particularly if the mRNA, the gene that's been expressed, is important for cellular function. So let's concentrate initially then on the first of these examples, really, the mutations which are introduced during DNA replication. So this is one of the major causes of errors and DNA damage, if you like. So the first thing to realise is that the fidelity of DNA replication itself is actually quite staggering. So the actual fidelity of DNA replication is 10 to the 10. So what that means is that every time DNA replication occurs, 1 out of every 10 to the 10 bases will be incorrectly incorporated. So 10 to the 10 is 10 billion. So if you think about the size of the human genome, which you should know is around 4 billion base pairs, it generally means every time that those 4 million base pairs are copied, there isn't generally an error made. Yeah? There's only an error made every 10 billion. So the actual fidelity which exists is high enough to ensure that DNA replication occurs perfectly during the cell cycle. So how does that happen exactly? Well, one of the things that's interesting is if you look at the actual fidelity of the enzyme which copies the DNA polymerase, the fidelity of that enzyme is actually only 10 to the 5. So what that means is every 10,000 base pairs that DNA polymerase copies during replication, it will make one mistake. So if the enzyme that's doing the copying makes a mistake every 10,000 base pairs, how on earth can it be the case that, sorry, not 10,000, 100,000 base pairs? How is the fidelity increased to 10 billion for the actual process overall? Well, the first thing that's actually important to realise is that while DNA polymerase itself has this 10 to the 5 fidelity, this is actually in real life more like a 10 to the 7 fidelity, and it's increased in accuracy due to this process called proofreading, which I'm not going to go into detail of now, other than to briefly explain what it is. So proofreading refers to an innate enzymic activity which DNA polymerase possesses, whereby it can actually repair itself an error that's been made during replication. So if DNA polymerase itself spots that the wrong thing has been incorporated, it's able to activate 3' to 5' exonuclease activity and kind of put the enzyme into reverse back along the DNA it's just copied, removing the incorrect base, and then it puts it back into first gear and off it goes again, replicating the DNA. So this proofreading activity is kind of like a double checking of the DNA that the enzyme's copying. It kind of double checks itself, and if it spots an error, it can go into reverse, go into this 3' to 5' exonuclease activity to remove that error, and then carry on in a 5' to 3' manner, replicating the DNA again. So this exonuclease activity, this proofreading, actually affords the process a 10 to the 7 fidelity. But how do we actually get to this 10 to the 10? So there's another 3 orders of magnitude here to make up. This is still only 10 million. One mistake every 10 million, which isn't good enough when there's 4 billion bases to accurately copy. So this final level of control is actually given to the cell via this mismatch repair system. So this is a DNA damage repair system which exists for the purpose of making sure DNA replication occurs with extremely high fidelity in the cells. And much of his understanding for how this process works is derived from E. coli. So that's where the work delineating and understanding how this mismatch repair system operates was originally done. So in order to actually function as a post-replicative repair system, the mismatch repair system needs to be able to do several things. So the first thing, obviously, it needs to be able to do is recognise when a base pair has been incorrectly incorporated. The second thing it needs to be able to do then, and this is very important, it needs to be able to determine which base is actually wrong. So if you think back to the example earlier on where we ended up with a C opposite an A rather than a T opposite the A, once the mismatch repair systems recognise that there's an error at that point, it needs to know whether the C is incorrect and shouldn't be there, or whether the A is incorrect and shouldn't be there. Clearly you should never have a C opposite an A, a cytosine opposite an adenine, because A's always go opposite T's and C's always go opposite G's. But with regards to whether it's the A that's wrong or the C that's wrong, how does the system know that? And the final thing it needs to be able to do is obviously repair the base. It needs to get rid of the wrong base and put the correct base in its place. So how does this work in E. coli? Well, it works via 3 proteins really. It's quite an elegant, simple system. The first protein, which is a dimer, is Mutagens, and what Mutagens does is it moves along the DNA recognising where an error in DNA replication has occurred. And it recognises this error by virtue of the fact that when you misincorporate base during DNA replication, so when you have an A opposite a C, for example, it changes very slightly the actual structure of the DNA. So the double helix looks slightly different, and this difference is detected by the Mutagens dimer, which then binds to this alteration, to this site of the DNA where this alteration exists. Once it binds then, Mutagens recruits a second protein called Mutel. And what Mutel does is it allows the actual recognition of this final piece of the jigsaw, this MutH protein. And MutH is kind of the clever part of the system, if you like. So once you have all these 3 proteins come together, the actual DNA strand where the error has occurred is nicked here. The DNA is resected back, it's chewed away in this error strand, past the point where the error has occurred, and then DNA polymerase comes along and it recopies this information again from the template strand, hopefully this time not introducing the error that was there previously. So MutS comes along, it recognises the error, binds with MutL, which in turn then leads to the recruitment of this MutH protein. It's MutH which is able to actually nick the DNA. So if you like, break the DNA strand on which the error has occurred. The DNA strand on which the error has occurred is then chewed up by our exonuclease activity, past the point where the actual mismatch has occurred, and then DNA polymerase comes along and just recopies this information correctly, repairing the DNA. So that's how the repair works, but how does the system know to actually get rid of this strand? How does it know that this strand is where the error has occurred and not this one? So if the error has occurred during replication, then it's been misincorporated on the daughter strand. In other words, the correct information which exists on the template strand has not been accurately copied onto the daughter strand by DNA polymerase. So how actually this system works in E. coli is via something called hemimethylation. So as you may or may not know, E. coli DNA is interesting because it's methylated by an enzyme called DAM methylase in a very specific manner. So every time that you encounter a specific sequence of DNA, GATC, in the E. coli genome, you'll find that it's methylated at that point. So this methylation occurs every 264 base pairs, roughly, in the genome. However, when you replicate E. coli DNA, the replicated daughter strand, the new daughter strand here, isn't methylated immediately by this DAM methylase enzyme. It takes the DAM methylase a little while to catch up with the replication machinery and start methylating the DNA. So what that means is when the repair machinery comes along, it's able to discern which strand is the actual parental strand and which is the new strand, because the parental strand will be methylated but the new strand will not. So it knows that the error must be on the new strand, so it gets rid of the new strand, the strand that isn't methylated. So methylation in this instance can be thought of as like a memory sensor, allowing the machinery to know which strand it needs to get rid of. How this strand's actually chewed up, it depends on where this break occurs. Sometimes MUT-H will bind downstream of the error, in which case it needs to be resected in a 3' to 5' manner. In that instance, exonuclease 7 or REC-J is the enzyme which chews back the DNA. If the actual break occurs 5' of the error, then a different exonuclease is used, exonuclease 1, and that chews away the daughter strand up until and past the point where the error has occurred. So the mismatch repair system is also present in humans. It's a very important system in humans, and it functions in a very similar way. There are a lot more proteins involved in the process in humans. It's a little more complex, but the basics of it remain the same. The MUT-S protein, this dimer which recognises the error in E. coli, is essentially made up of a family of proteins called the MSH proteins in humans. The MUT-L proteins made up of the MLH proteins here. We know that it's a very important system actually because a particular type of familial colon cancer is associated with mutations in this MSH2 gene here. So if you mutate this gene, which is responsible for repairing DNA in this way, you become incredibly prone to colon cancer, highlighting how important this system is for maintaining genomic integrity and not introducing mutations which might lead to cancer and cellular transformation. One interesting difference, however, is the fact that in humans we lack a MUT-H homologue, so we don't have this clever MUT-H enzyme which recognises which is a new strand. One of the reasons we don't have this enzyme probably is that our DNA isn't like E. coli's, it isn't hemimethylated. That's a process unique to that actual bacteria. So in the absence of that memory system, how does the mutation mismatch repair system know which strand to get rid of in humans? Well, people think one explanation might be given by the Ozaki fragments. So the presence of Ozaki fragments might inform the mismatch repair system that this is actually the daughter strand, the new strand here. But that wouldn't explain how the actual leading strand during DNA replication is recognised, because this doesn't fragment into these fragments, obviously, these Ozaki fragments. Current research suggests that it's actually all to do with this very special enzyme, which we'll talk more about next year, at the replication fork called PCNA, which is a processivity enzyme, and it's thought that the actual repair system proteins associate with this processivity enzyme during replication, and by associating with that know which strand to actually repair during mismatch repair. But that's still very much, the jury's out on that research-wise. So thus far we've looked at natural things really that occur during DNA replication, so the errors that occur during the replication of the DNA and how they're repaired. But what about more unnatural changes to DNA, if you like? So I mentioned at the start that when you talk about unnatural errors in DNA damage agents, you probably start thinking of radiation. I think that's one of the big ones that people go for, just because obviously historically Marie Curie, cancer, and the Incredible Hulk. Seems a bit blasphemous talking about the Incredible Hulk in the same sentence as Marie Curie, but you get the idea. Radiation's a well-recognised mutagen of DNA. However, what you might not know is it's actually two things we require for life: oxygen and water, that present a much greater risk actually to the integrity of your DNA. During your life, it's far more likely that you'll end up with a mutation that will give you cancer by virtue of water or oxygen than it is from ionising radiation. And one of the reasons it's more likely is that there's no avoiding this sort of intrinsic DNA damage. So reactive oxygen species and obviously water are just byproducts of physiological processes. You can't avoid them, but they're both in their own way very damaging to the DNA molecule. So how are they damaging? Well, one of the ways in which they're damaging is via deamination. So deamination effectively represents a major threat to DNA integrity, and the most common form of deamination that occurs in your DNA is the conversion of a cytosine base to a uracil base. And this occurs via the action of water. So this is a hydrolytic attack on the DNA by water molecules, which alters the structure of cytosine in such a way that it becomes a uracil. So effectively you end up with an amine group being removed from this molecule, which gives you a uracil molecule. And obviously cytosines in DNA are generally found, well, always found opposite to guanine, a G. So C is found opposite a G. A uracil, which occurs in mRNA instead of thymines, is always found opposite an A. So as soon as you have a deamination event of this type, you start introducing the possibility of mutation into your DNA sequence. So just to reiterate, adenine rather than guanine will be introduced opposite the strand during replication. That's the problem of this deamination event. And it probably interestingly explains why DNA contains thymine rather than uracil. So for a long time scientists didn't really understand why that was the case. Energetically it makes a lot more sense actually for DNA to contain a uracil molecule than a thymine. So thymine is complementary to an adenine. So T's are found opposite A's in DNA. Also, uracils are found opposite A's in mRNA. So the uracil can quite happily do the job of a thymine with regards to maintaining that information in the nucleic acid sequence. In fact, energetically, it's better at it. But the body instead chooses to incorporate thymine into its DNA, a less energetically sensible decision, which is unusual in biology. But it turns out, once we started to understand about deamination, that there's a very good reason for this. That reason being. The reason being that if the body actually incorporated uracil into the DNA sequence rather than thymine, there would be no way for the machinery that goes about repairing DNA to tell whether the uracil present in the DNA sequence was there because it's supposed to be there, or whether it's there because it's there after a deamination event has occurred to a cytosine base. So it's a really interesting example of how finding out more about molecular systems has answered a bit of a global, if you like, or a bit of an enigma, a mystery that's existed for a long time in regards to the actual makeup of DNA itself. So it's not just cytosines that undergo deamination. You can get deamination of adenine and guanine as well. However, this is a lot rarer. And you can get very severe forms of hydrolytic attack, which result in depurination. So this is the actual removal in its entirety of a purine base, an A or a G, from the DNA backbone. That obviously results in a small deletions to the DNA, which can introduce frameshift mutations and cause major problems with the function of proteins that have been translated from that gene. It's normally relatively straightforward for the cell to recognise whether or not a deamination event's occurred. It's pretty obvious if a uracil appears in the DNA sequence, it shouldn't be there and it needs to be repaired. However, there are certain situations which prove more problematic with regards to recognising when errors have occurred. And a good example of that is the actual deamination of a specific type of DNA base called 5-methylcytosine. So these methylated cytosine bases are often found upstream of genes and act as transcriptional silencers. However, unfortunately, if you actually deaminate, if one of these 5-methylcytosines undergoes deamination, it gets chemically converted into a thymine molecule. So we've got a situation here where 5-methylcytosines are found all over the genome, they're perfectly normal, they help repress gene expression under certain circumstances. But if they're deaminated, they're converted to a thymine. Obviously, thymine bases are also perfectly normal, found all over the genome. So it's very difficult to actually spot when actually this event's occurred and to repair it accurately. And for that reason, these 5-methylcytosine bases are actually hotspots for mutations throughout the genome. So the second type of intrinsic attack then is from reactive oxygen species. So you probably remember from A-level chemistry, if you took it, that these ROS, these reactive oxygen species, are very damaging oxidising agents, and they actually represent a major source of DNA damage. So they're produced via a variety of methods, ionising radiation being one, but also as a consequence actually of cell metabolism. So we can't escape these things. These ROS, these reactive oxygen species, are flying around in the cells all the time. In fact, the cell's got all sorts of mechanisms for dealing with them. But occasionally, reactive oxygen species will be present at such a level or in such a way that they can damage the DNA. The most common way that they actually damage DNA is via oxidation of a guanine, leading to the formation of a 7,8 dihydroate oxoguanine, which is a bit of a mouthful, or OXO-G for short. So OXO-G, it's not gravy, you don't put it on your Sunday dinner, it's just a shortened term for this longer oxidated version of guanine. The problem with these OXO-G bases, these oxidated guanines, is that they're promiscuous with regards to what they pair with. Normally a guanine will only pair with a cytosine, a G will only be found opposite a C. However, OXO-G will just as happily pair with an adenine as a cytosine. So again, you're introducing these sorts of mispairing events which eventually can lead to mutations. So in this instance, a G pairing opposite an A will eventually lead to a T to A transversion mutation, were this to go undetected and DNA replication to proceed through a couple more generations, as we saw at the beginning of the lecture. So they're intrinsic DNA damaging agents then. What about extrinsic agents? So these are now the mutagenic chemicals, etc., that we know are able to damage our DNA. How we know they damage DNA, one of the classical ways in which people would test and still to a certain extent test chemicals to see if they're DNA mutagens is via something called the Ames test. So this is where you effectively add the chemical to a particular type of bacteria. I forget the actual type now. I think it might be Bacillus, but I'm not sure. Or Salmonella, one of the two. I think it's Salmonella. But the point is, you grow Salmonella in culture, and this Salmonella requires histidine to be present in the growth media in order for it to grow. So it requires histidine to be supplemented in that media. However, if you grow these bacteria on agar plates lacking histidine, what you will see is that one or two cells will end up growing as colonies on those agar plates, for the simple reason that the mutation rate in bacteria is relatively high, and you're growing a large amount of cells obviously in culture. So of those billions of cells which are present in culture, one or two will revert in such a way that the mutation in histidine, which means that the cells require it to be supplemented, is reverted back to wild type. So you'll get these revertants appearing on the plates. One or two colonies will grow. If you add a chemical to these cells during culture, which is a known mutagen, or turns out to be a mutagen, the number of revertants you see will be increased because the mutation rate of the bacteria is increased by virtue of the addition of that chemical, which leads to more revertants appearing on the agar plate. So the Ames test is just a good way of detecting whether or not a chemical is a mutagen or not. One such type of mutagen are these alkylation chemicals, and what these chemicals do is they transfer specific groups to the bases in the DNA, or indeed to the phosphate backbone of the DNA itself. And these different modifications can be methylation, ethyl groups, etc. And the consequence of placing these different chemical groups onto the bases is that you affect the promiscuity of how that base then interacts with its partner. So the best example of this is actually the alkylation of an alkylation event which occurs to the thymine base, the guanine again base pair, in such a way that you had a methyl group, had a methyl group to the guanine, which results in it again being promiscuous and being able to pair with thymine as well as with cytosine. So this is highlighted in this diagram here. So in effect, you take guanine, you methylate it at the 6th carbon, in such a way that it then becomes promiscuous with regards to the base pair. It becomes promiscuous with regards to the other base pairs it can bind with. There are other examples as well. Another important group of chemicals are base analogues and intercalating agents, and these are really the other two major type of mutagens that exist. One example, or the best example of a base analogue, is something called 5-bromouracil. So base analogues are what the name suggests. These are chemicals which look like, smell like and taste like normal nucleotides, normal bases. However, they're subtly different in such a way that they can be catastrophic to the cell were they to be incorporated by mistake. So if you were to include 5-bromouracil into your cell culture when your cells are undergoing replication, it's just as likely that this base analogue will be incorporated in the new daughter DNA as the actual base thymine. Unfortunately, fibrillum uracil, unlike thymine, is very promiscuous with regards to base pairing again, so it can lead to introduction of mutations. Intercalating agents are slightly different. So these are very flat compounds which are actually able to slide between the bases which appear in the DNA double helix in such a way that it leads to issues during DNA replication. And the best example of this is ethidium. So ethidium bromide is used in the lab to detect DNA when you run DNA on gels. We talked about this in a lecture last week on PCR. It's a chemical which intercalates, which binds to the DNA. In the context of this lecture, we want to think about how that might be a problem for the cell. And the way it's a problem for the cell is that it affects DNA replication, as we'll see later on. So we've talked there a little bit about how the DNA itself is damaged, but how is all this damage actually repaired? So how are these mutagenic bases recognised and removed by the cell during replication, just during cell division in general? Well, there are several mechanisms which exist. I'm just going to talk you through those now. The first of those is base excision repair, and it's generally the most simple. So it works on the premise that you need to recognise, remove and then replace the damaged base. In the case of base excision repair, this mechanism relies on a specific group or family of enzymes called glycosylases. And glycosylases exist to remove specific mutations which have occurred or DNA damage events which have occurred in DNA. And they effectively come along, recognise a specific error, snip out the damaged base, and then DNA polymerase comes along and fills in the actual error to repair the DNA. So importantly, specific glycosylases exist for specific errors. For example, there's a specific glycosylase called uracil glycosylase, which is responsible for recognising deamination of cytosine to uracil, if you remember this common event that we talked about a few slides ago, in such a way that it's repaired properly. So there's the glycosylases which exist just to repair that type of damage. And in fact, there are several now. I think there's 8 or 9 independent glycosylases which have been identified in man, all of which go about the business of recognising and repairing one specific type of DNA error. Yes, so it's a very smart bomb, if you like, way of approaching repairing DNA damage. Obviously, uracil glycosylase can't go along and repair a deamination event that's occurred to something that isn't a cytosine. It doesn't work for something else, it just works for that specific event. So one of the interesting things about these glycosylases is how do they actually get access to this damaged base in the first place? So if you think about the base that needs repairing, it's buried inside the double helix of the DNA. So it's not easily accessible by these enzymes. It's not easily accessible by these enzymes. They can't just come along and cut this thing off easily and then replace it and wander on to the next job. But how do you get access to this damaged base when it's buried so deep in this double helix molecule? And this kind of was answered by some nice work using X-ray crystallography and NMR, so structurally looking at the DNA molecule itself. And scientists realised that yes, the DNA bases are kind of buried deeper than the double helix, but energetically it's very easy for this base to kind of flip out of this double helix so that it's no longer located on the interior but the exterior. So if you think about this as just rotating on this axis here, so this base flips, and then all of a sudden this base is very accessible then to be snipped off by this glycosylase and replaced with the correct copy. And then once the correct copy is there, it just kind of rotates back in. So it's a nice example of another facet of the structure of the DNA molecule, insofar as how it allows easy access to these bases for repair. And obviously speed is an issue during DNA repair, energy is an issue, ATP consumption. So you want to be able to do something energetically cheaply but also swiftly, and this is a really nice explanation for how that actually occurs during the action of these glycosylase enzymes. So excision repair is the most common mechanism for repairing DNA damage, but there are actually simpler mechanisms than that. So direct reversal effectively just undoes the damage that's been done in the first place without the need to replace anything. One example of direct reversal, a good example, is reversing the type of damage that occurs during UV radiation. So this is another really major source of DNA damage. So if you like to cook yourself on a sunbed like this guy does, or you're like a student that we had in the labs during final year projects a few years ago now, who likes to use the UV source without a safety mask, you'll probably end up with skin that looks like this and being quite sore for a number of days. A little bit more worryingly, you may actually introduce permanent damage to the DNA in your skin, and which in turn can lead to skin cancer. And that occurs via a specific type of DNA damage called thymidine dimers. So this is where two adjacent thymine molecules, bases within the DNA molecule, effectively become fused together, joined together by a cyclobutane ring here. And what this means is that these two bases can no longer actually complementary pair properly during DNA replication, and you can get frameshift mutations being introduced due to the failure of DNA polymerase to copy these base pairs properly. Now, there's a very simple, elegant system that the cell uses to get rid of these thymidine dimers, and that's via an enzyme called photolyase. Photolyase comes along and uses the energy from natural sunlight, from natural light, to actually break these two bonds here, which releases the two thymines and gives off energy which photolyase then incorporates, and it can go back doing the process again. So it's energetically very cheap. It's specific to this particular type of damage, and it, rather than chopping it out and replacing the actual damaged DNA bases, it actually just repairs them directly. There's another example of this, which is how the cell goes about repairing this methylated guanine we talked about earlier on, which can arise from alkylating agents. So if we look at this guanine here, it's got a methyl group placed on it. And remember, these methylated guanines are promiscuous, they can base pair incorrectly. So what the cell can do actually is just remove this methylated group here using a methyl transferase enzyme. So this methyl transferase enzyme will come along, transfer the methyl group from the guanine to itself, and then go off again. Now, energetically, this is very expensive, and the reason it's expensive is this is a one-step process. This can't be recycled and then go do the job again. So you need one molecule of this methyl transferase for every removal of CH3 that you want to carry out on a guanine. So it's not ideal for the cell to do this, but it will do this if needed, because it's less ideal, obviously, to start mutating and introducing genomic instability to the cell. So there's also a more complex system that exists for repairing damage to DNA, and this is called a nucleotide excision repair system. So what we've talked about thus far are repair mechanisms which exist to effectively recognise and repair very specific changes to the DNA, one or two nucleotides that have been altered due to DNA damage events. Nucleotide excision repair is not really interested in detecting specific changes, it will just detect errors of any type that exist in the DNA double helix. And in E. coli, this nucleotide excision repair system is effectively directed by 4 different proteins: UVRA, UVRB, UVRC and UVRD. Now the UV radiation prefix to those genes kind of gives you an indication of the fact that these genes are actually important in also repairing damage caused by ultraviolet light. And in fact, if you start mutating the human homologs of these genes, it can lead to diseases which are associated with extreme sensitivity to sunlight and predisposition to formation of skin cancer, etc. So how does this system work? Well, it works in E. coli by recognising damage, again by virtue of a change in the actual structure of the DNA double helix. So UVRA, which is in a complex with UVRB, scans along the actual DNA until it recognises a part of the double helix where the shape's wrong, which is indicative of a mutation present in the DNA bases. UVRB, which is a DNA helicase, then unwinds that region where there's an error, and UVRC is recruited, which is an endonuclease that will nick and cut the DNA where the errors occurred. But a second DNA helicase called UVRD then comes along and starts unwinding the entire double-stranded helix to give access to this part of the DNA where the errors occurred. Some more endonucleases will come along and remove that region which contains the error, and the DNA polymerase will repair the error using the DNA template strand, the other strand as a template, in such a way that it's then completely repaired and ligase comes along and joins a phosphate backbone again. So the actual nicking of the DNA here, the chopping of the DNA, is very specific. In E. coli, it occurs 8 base pairs upstream of where the error is, and then also 4 or 5 base pairs downstream of where the error is. So you remove about a 12 base pair region from the actual damaged DNA strand, and that 12 base pair regions are then repaired using DNA polymerase and DNA ligase. In humans, there's another Strikingly similar system to what exists in E. coli. A lot of the genes from E. coli have homologs in humans, XPC, XPA, XPD, etc. This time the XP prefix refers to xeroderma pigmentosa. If you remember from human genetics last year, we showed you that horrific picture of that poor girl who had skin cancers all over her face. It's an example of genetic heterogeneity. The idea that different mutations in the same gene, mutations in different genes can give rise to the same kind of disease. And mutations in these different XP genes are what gave rise to the actual disease xeroderma pigmentosa in that young girl. So I told you at the time that these XP genes function in a pathway, and that pathway is this nucleotide excision repair pathway. So these genes here, the homologs of the genes in bacteria, obviously the system in humans is a lot more complex. There's actually almost 30 proteins, around 30 proteins now that have been identified that function in nucleotide excision repairs compared with 4 in E. coli. So it's a very complex system in humans. One of the major differences is the size of the patch, if you like, that's removed from the damaged strand in humans. If you remember in E. coli, I told you it's between 12 and 13 base pairs. In humans, it's around 23 to 32 nucleotides that are removed. So there's a larger region taken away and repaired during the process. Really, you can think of this nucleotide excision repair system as being almost like molecular health and safety officers for DNA. So they go about the business of just scanning everything to looking for mistakes that might be present, mistakes that maybe have been missed by other systems. And in humans, it's important to realise that this nucleotide excision repair system comes in one of 2 flavours. So it can either be global genomic NER or transcription coupled NER. Global genomic NER basically refers to the fact that this system is able to just seek out damage to DNA on a global basis. It's not really understood how the nucleotide excision repair machinery is able to be directed to just locations in the genome. Seems unfeasible, but this protein complex can just scan all 4 billion base pairs continually throughout the genome looking for mistakes. So it's likely that DNA damage sensors are important here. So these are proteins which will be recruited to damaged areas of DNA and then give off, if you like, a signal or provide a signal to the nucleotide excision repair machinery saying, look, there's a problem here, come along and sort it out. The other type of NER that exists is transcription coupled NER, and this kind of works on a signal basis as well. It's a slightly more direct and ordered process than what must exist for global genomic NER. So transcription coupled NER basically is important to realise it acts specifically on genes which are being transcribed, actively transcribed. So the first thing you should realise is a way of prioritising DNA damage to those parts of DNA which have been actually used by the cell at this point, which have been transcribed into mRNA and made into protein. And it works by virtue of the stalling of this RNA polymerase II enzyme, this enzyme which goes around copying DNA into mRNA during transcription, gene expression. So what happens is the RNA polymerase holoenzyme, this big RNA polymerase multi-protein complex which goes around copying DNA into RNA during transcription, the red arrow here represents the mRNA molecule. If it reaches a point in the DNA sequence here where there's a structural problem, a change in the DNA sequence because of DNA damage, it will stall, it will stop. And when it stops, this is recognised by some of the enzymes associated with this RNA polymerase. And those enzymes go about giving off a signal telling the nucleotide excision repair proteins that they are needed at this point to repair some damage. These NAR proteins are then recruited, partly by virtue of a RNA polymerase II associated protein called TF2H, which we'll talk about in a lecture later in this module. And these proteins are recruited, a large number, upwards of 30 of them, to this region, and they get rid of about 30 nucleotides around the point where the damage is, and then these 30 nucleotides eventually repaired via DNA polymerase in such a way that that error is removed. So moving on then, what we've talked about thus far is repairing damage via the cursity or the use of an undamaged complementary strand. So in each case we've talked about DNA polymerase coming along and repairing the error. It's only been able to do that because there's a parental strand present which hasn't got an error in it, which hasn't been damaged. So that acts as a template for the repair machinery to come along and basically correct the mistake. Yeah. So there's a master copy there, yeah, which can be used to make sure that the new copy is correct. What happens if that master copy isn't present? So what does the cell do then? And this is a real common problem really in DNA damage circles for the cell, because the event which leads to this loss of template strand, in other words these double strand breaks, is relatively frequent throughout the life of a cell. So there's a couple of ways in which double strand breaks can actually be repaired. The first is via homologous recombination. So homologous recombination is a really important process. We touched upon it in human genetics last year when we talked about meiosis. I'm only going to mention it now because whilst it's important in bacteria for repairing double strand breaks, homologous recombination, it's not as important actually in humans. We use a slightly different system that we'll talk about in a second. Effectively in homologous recombination, what happens is where a double strand break has occurred in the DNA and therefore you have lost information from both the new and the template strand, what the cell then does is it uses the fact that there is a second chromosome in the cell which contains that DNA information, which can be used as a template to repair the actual chromosome where the double strand break has occurred. And it can be used via this homologous recombination process. And I would suggest you read about this in the core text in a little bit more detail, because in effect you could devote a lecture to this in itself. I'm not suggesting you need to go away and learn an entire chapter on homologous recombination, but you need to have a good and basic understanding of what the process is. You need to be able to describe what's going on in this diagram in effect. So what happens is, following double strand break repair, one of the strands from the broken chromosome invades and displaces the DNA from the actual undamaged chromosome copy in such a way that you get this bubble form. And then this displaced strand is able then to use information in the homologous chromosome to copy and repair the information that's been lost during this double strand break process. The same is true of this displaced strand from the undamaged chromosome, which can be used as a template for repairing the other strand in the double strand broken chromosome, as shown here. And the upshot of that is, once these two strands have been used as templates, you can separate the two joined chromosomes apart by nicking the DNA here, and what that means is you've used the information in this chromosome to repair the other. However, by virtue of doing recombination repair in this manner, and depending on how you separate these two joined strands, whether you cut them this way or this way, it can affect the type of recombination, the sharing of information that goes on between the two chromosomes. So it's this recombination event here which allows you to mix genes between germ cells during meiosis, and it also allows you to repair double strand breaks that have occurred in chromosomes, in bacteria primarily, but also occasionally in humans. So what happens then if you've not got a second copy of the chromosome present in the cell? So in other words, what happens if a double strand break occurs prior to actual DNA replication occurring? And this is actually resolved via a process called non-homologous end joining, and this is a process which is more common in humans, human cells, as a way of dealing with these double strand breaks that occur. It doesn't actually occur in all bacteria, doesn't occur in E. coli, non-homologous end joining. It has been shown recently to occur in bacillus, but it's generally a mechanism of eukaryotic organisms. It's quite problematic for the cell to use non-homologous end joining, because effectively what you're doing is you're introducing bases at random, so to speak, to repair damage. So it's easy to introduce errors in this way, and it's one of the reasons why double strand breaks are so problematic for chromosomes and for cells. Such a normally a catastrophic event. What happens during non-homologous end joining is a family of proteins called Q come along and bind to the double strand break repair. These Q proteins then recruit a whole family of other proteins which are responsible for initially chewing back further the DNA around the double strand break before then copying new information across this double strand break in such a way that the break's repaired in the actual double stranded DNA molecule. So finally, just to finish, there is a if all else fails mechanism which exists in DNA damage, and that's this translesion repair mechanism. So if everything else has gone wrong, if the exonuclease activity of DNA polymerase hasn't worked, hasn't been able to repair the error, if the mismatch repair system's not worked, if the glycosylases have failed to spot the error, and the error still remains, then you end up in a situation where the DNA polymerase enzyme wants to copy a piece of DNA sequence, but it can't get past this adduct, this error which is present in the DNA. In this instance, it's indicated by this thymidine dimer. So in that instance, the cell's got a choice to make. It can either not replicate fully a chromosome, which should be catastrophic. If you don't completely replicate a chromosome, the cell's just going to die, or it's going to have severe genomic instability. Or it can just say, look, I know there's a problem, but just ignore it, copy the information anyway, and don't worry about the fact that it's likely to be wrong. And that's effectively what goes on in translesion DNA synthesis. So DNA polymerase will reach such an impasse, such an error in the DNA which it can't get past, and it will be replaced by a different type of DNA polymerase called a Y DNA polymerase. So this is a specific family of DNA polymerases that are template dependent. They still need a template to be present in order to copy the new strand, but they're not base pairing dependent. So they're not bothered about copying the right thing opposite the error. They'll just use any nucleotides they can to get past this structural damage to the DNA. So obviously, because of that, the propensity for introducing errors is high. These things have got a poor fidelity rating, but it's better to copy something incorrect than not copy it at all, because not copying completely a chromosome will definitely be a catastrophic event to the cell. Obviously it makes sense that these Y polymerases aren't just kicking around in the cell during DNA replication. You wouldn't want them accidentally starting to replicate parts of the genome because they're so promiscuous with regards to what bases they add. You know, they're very inaccurate, and for that reason they're actually not expressed in E. coli at all, except for in circumstances where the DNA is known to be damaged, and this is what's termed the SOS response. So these Y polymerases only become expressed when DNA damage has been detected, so that they're there ready to do this translesion synthesis if needed. So just to summarise then what we've been talking about today, we've been talking about the damage that occurs to DNA via DNA replication, or direct attack from numerous factors such as intrinsic factors which are unavoidable, like attack by reactive oxygen species and water. Actually it's hydrolytic deamination which is one of the major causes of DNA damage, this deamination of cytosine to uracil. But there are also other DNA damage events which occur via alkylation chemicals and base analogues, intercalating agents, which are also important. Ionizing radiation is a problem because it causes double strand breaks, which are very difficult to repair. But double strand breaks, in addition to all of these different types of misincorporated bases or modified bases, can be repaired by the cell using a variety of different mechanisms which have evolved in such a way to ensure that when we replicate our 4 billion base pairs, ideally none, or certainly no more than 1 or 2, are actually altered during each round of division. Yeah. So I'd like to leave it there. So hopefully this podcast will be a useful revision aid, like the other podcasts in the module, when you come to start reading over or looking over this material again. I'll emphasise again at the end of the lecture how good the molecular biology of the gene recommended textbook is for molecular biology. It's also a level 3 appropriate textbook, so if you were to purchase it, you'd be buying a book that will be useful certainly this year for molecular genetics, but also for, I'm sure, a number of modules next year as well. It's a great molecular biology textbook. Okay then, so that's the end of this lecture. I'll see you again for the next lecture in the podcast series. Cheers. I won't, there's one more slide. So this is just a table which you might find useful that kind of summarises the different types of repair mechanisms which exist depending on the DNA damage event which has occurred. So I'll leave it with that. Thank you. Bye bye.